Good afternoon, and welcome to the second annual Duke University Center of Exemplary Mentoring Research Summit. My name is Calvin Howell. I'm a professor of physics here at Duke University and co-PI with Paula McLean, Dean of the Graduate School, and James B. Duke, Distinguished Professor of Political Science, on the award from the Alpha P. Sloan Foundation that supports the Duke University Center of Exemplary Mentoring, referred to as UCEM. The UCEM is a collaboration between the graduate school and 9 departments in the physical sciences and engineering. Its overarching goal is to work toward increasing the diversity of our campus research community by recruiting and supporting students from underrepresented communities in the US in PhD STEM fields. The focus of UCEM activities is on mentorship, primarily because of the apprenticeship nature of graduate education. Foundational to these efforts is building a community that supports our Sloan Scholars, that spans across disciplinary boundaries and over department walls. Traditions are an important part of building and sustaining a vibrant academic community. We are delighted that you have chosen to participate in the second annual UCEM Research Summit and to be part of establishing this summit as one of Duke's traditions. The Research Summit provides an opportunity for our scholars to come together as a community to learn about the research questions that drive discovery for Sloan Scholars. You're here today from students who are in the second and third years of their PhD programs. At its core, the summit is driven by the idea that community and collaboration are central to what it means to be a scientist. This summit gives our Sloan Scholars an opportunity to introduce themselves to the broader Duke community through sharing some of their research interests. Again, thank you for attending this year's summit. I'll now turn things over to the director of the Duke UCEM, Jacqueline Looney, Senior Associate Dean and Associate Vice Provost for Academic Diversity. Dean Looney will introduce our scholar speakers today. Thank you.
Thank you, Professor Howell. First, I want to start by thanking everyone for joining us today. We know that you're probably all Zoomed out, but what better way to Zoom in to see our scholars do what they do best, talk about their research. I want to especially thank the faculty members who have joined us. For you play the most vital role in shepherding the scholars through their programs, and we are so grateful for that. And then our scholars for all the hard work that they're doing and progressing. We have 30 scholars in this program, and all scholars are on track and doing exactly what we expected them to do when they entered Duke. This second annual research summit brings the Duke UCEM, as we call it, full circle with the first grant. The scholars presenting today have been preparing for the last 6 weeks with coaching from Assistant Dean Melissa Bostrom and the Sloan grad interns Dena Zhu Ho and Zane Swanson. During this program, the scholars have been asked to present a talk on their research question in 4 minutes. Later in the program, we have set up breakout rooms so that you can talk with the Sloan Scholars and learn more about their work. This will follow Dean McLean's remarks. So first up is Adriana Stone, Electrical and Computer Engineering.
Hello, my name is Adriana Stone. I am a second-year PhD student here at Duke. I study optical engineering and imaging science, and today I will be presenting imaging through foggy barriers. If you're not familiar with imaging science, I'd like you to know that it is an enabling technology. We build cameras and design imaging protocols to help other scientists do their science better by taking their eyes places they weren't able to go before. We operate in fields like biology, medicine, astronomy, defense, or really anywhere a picture is being taken. I would also like you to know, or perhaps just remember, that we can store images as a matrix of numbers. So here I just have an image of a plus sign, and on the right-hand side of my slide, I have a matrix of numbers containing the exact same information, just with numbers. And so it's in this way that we can store any image as a matrix of numbers. And so this can permit us to do basic math operations like addition or subtraction, but it also means that we can incorporate higher knowledge domains in our image analysis, like statistics, signal processing, or physics. And this is where the bulk of the heavy lifting gets done, is in image analysis, because we can use math and other things like statistics or signal processing to extract more information from our images than what we would be able to see with our eyes. Well, let's return to the problem of imaging through foggy barriers. You're familiar with this problem if you've ever looked through frosted glass. What happens is as the light passes through the glass, it becomes scattered, scrambled, and all mixed up, and kind of gives it like a frosty or foggy appearance. And we as image scientists would like to study this problem and develop an imaging protocol that would allow us to image through frosted glass as if the frostiness weren't there. And so to recreate this problem in the lab, we use something called ground glass, which is glass that has been ground on one or both sides with sand to give it a frosted look. And so let's have an experiment. My experiment is here at the bottom of my slide. There's a laser that's lasing light on an object. And here it's just a simple letter E, where the E shape is cut out and light can pass through. And then that light goes through ground glass where it gets scattered and scrambled. And then we take a picture of that scattered light. And that's the picture in the middle of my slide. It doesn't look good. You cannot look at this picture and tell me that we were trying to image a letter E. But what's interesting is that there's actually a hidden structure and information in this image. And we can take advantage of something called the optical angular memory effect and do some math and essentially do an image reconstruction and take my center image and move it and manipulate it until it becomes image on the right-hand side of my slide. And it looks pretty good. It looks like a letter E. But if we're being honest with ourselves, there's a little something to be desired, and it could definitely be a better image of an E. And so that's what my research focuses on, is how can we make these reconstructions better and better? And so the immediate utility of this technology is in microscopes. So when a scientist is putting tissue on a microscope slide, they have to be very careful not to put any extra tissue, because any extra tissue will have like a frosted glass effect and will scatter the light and make their image blurry and just not good. And so we can help them by applying this technology so that they can actually image what they were trying to image. And then you could also use this technology to literally just image through frosted glass or any other kind of foggy barrier.
Thank you, Adriana. That was great. Next up, Jessica Lalonde, Mechanical Engineering.
Thank you so much. Hi, everybody. Thank you so much, Dean Looney. Thank you to everyone for your time and for coming today. My name is Jessica Lalonde, and I'm a second-year PhD student studying materials engineering. My advisor here at Duke is Dr. Jennifer West, and I performed the work for this project with two senior mentors at Los Alamos National Laboratory as part of an ongoing research fellowship that I have with the Laboratory for Improving Sustainable Biomaterials. So what my research project is addressing is building a way to predict how plastic alternatives break down in different kinds of dynamic, ever-changing environments. We all know that the plastic problem is a global problem that has spanned multiple generations. But this type of approach and this particular project can help make a major impact on plastic pollution by helping researchers improve the way we design long-term plastic alternatives and the overall materials design process. Creating more effective plastic alternatives is an important part of the solution to move towards a future sustainability-focused materials economy. Polyhydroxyalkanoates, or PHAs, are a very large and diverse class of materials, but they are naturally derived bioplastics that have been extensively studied, and they offer a large chemical space for designing new types of materials. But right now, as it is, their degradation is currently not very well understood or extensively modeled. So that was my hypothesis here, is that we can use machine learning as a tool to learn from the data that we already have and predict how those really chemically diverse materials breakdown in the environment by learning from trends in previously published data to start to address the problem. First, we started by developing a database and accumulating a lot of data using already published and available studies for diverse PHAs and more traditional plastics like polystyrene or PET. In this way, the model really builds on many years, decades of open access data on PHAs. When I first made this database, I included features like polymer composition and experimental conditions for the degradation study, which then I could use to categorize the data via via a process called data curation. Using over 1,600 data points and about 190 samples, we built a machine learning model to fit the data with specific parameters and a formula according to a regression model, basically looking at how well the model fits that parameter and that formula. This model can in turn learn from the data to generate predictions on how the plastics in the database will break down over time. Ultimately, we were able to generate parity plots that look like this, to show how well our model is capable of predicting the different factors affecting degradation. These plots are essentially showing how the model is performing when we first train our model on some samples from the data, which are represented by the blue squares here. And then we show the model new unseen points, the orange circles here representing a test set, so we can look at how well the model is working. As you can see here, the test set of the data is lining up pretty well with the first set, with just the first parameter. But then when we expand the model to include a second parameter, we're finding that we're getting a much better prediction. So we can tell from these plots that our machine learning model is generating accurate and reliable predictions of the polymers within the database. In conclusion, we are currently able to generate parameters and weight loss prediction curves for the samples within our database, but this model doesn't really take into account the design trade-offs between the mechanical performance of the material and its degradability. The main areas of use for this particular model are currently in the prediction, research, and modeling of experimental blends of degradable PHAs, which would ideally assist in designing next-generation plastic alternatives for a healthier future ecosystem. Thank you very much for your time, and please don't hesitate to reach out later if you have any questions.
Thank you, Jessica Lalonde. That is great. Aitor, Brachio, physics.
Hi, everyone. So my name is Aitor Brachio, and I study particle physics here at Duke with the Phil Barbeau group. And we study what's called coherent elastic neutrino nucleus scattering, or we call it SEVNs for short. So as a quick introduction, what are neutrinos, right? So neutrinos are these electron-like fundamental particles, which basically means that you can't cut them apart, and they carry no charge. They're also the most abundant massive particle in the universe, but they're the least interactive. So that makes them really difficult to detect. In fact, most detectors don't measure them directly. If you look at the image on the right-hand side, you can see 3 distinct tracks. Those are 3 other particles that were generated by a neutrino collision. But prior to that, you can't see any neutrinos at all. This is proof. This is just showing that we know neutrinos exist. This was their first detection, but we can't see them directly. So as motivation for the research, we're very interested in studying neutrinos in particular because they have a lot of implications for radiation detection, and it goes so far as nuclear non-proliferation. So essentially, whenever there is any nuclear event, there is a uptick in the amount of neutrinos in the area. So we can detect them. We can know whether or not there is uranium enrichment going on, or if there is some amount of radiation leak at a radiation lab. For another thing, it improves our understanding of the standard model of particle physics, which is sort of our theoretical framework for how particles behave. And for another thing, it's just a new way to observe the universe. We can use this to look at, to study the birth of stars or the death of stars and such other things. So SEVNs itself was predicted over 40 years ago, and it's taken since then to detect it for the first time. Interestingly, though, it has huge cross sections relative to other neutrino events. So cross sections are a measure roughly of how probable an event is. If you look at the figure on the right-hand side, you can see all these lines, but there are 2 in particular that tower above all the others. These are the SEVNs events relative to all other neutrino events listed here. Their cross sections or their probability exceed the others by a factor of 10 at the minimum. This is also really well understood in the standard model, so it works as a really good test for the standard model. So why has it been so elusive? 40 years and we've never been able to see it up until now. So effectively, we've been smacking a bowling ball with a ping pong ball and trying to measure how much the bowling ball accelerates. It's very hard to see it. A neutrino is very tiny and discharges very little energy into the nucleus that it bounces off of. Those recoil energies are on the scale of keVs, whereas most detectors can't detect anything below 4 or 5 MeVs. So that's a thousand units of difference in like a scale of thousands difference. There we go. Sorry. So far, we have several detectors located at the SNS at the Oak Ridge National Laboratory, which is the image that was on the first slide. So we have all of these different detectors with different materials. And so far, they've been incredibly successful. We had first detection of sevens a few years ago, and we've managed to improve several parameters on the standard model itself. So in the future, we're hoping to construct many more of these detectors with new target materials. And we are hoping to reevaluate old neutrino data from before the sevens were ever seen for the first time. We also hope to collaborate with other institutions to help them calibrate their own versions of these detectors. And we're hoping to get more data acquisition and improve some parameter calculations on the standard model. With that, that's my time. So thank you, everyone. And I'm going to pass back over to Dean Mooney.
Thank you, Aitor. Gavin Gonzalez, Biomedical Engineering.
Hello, everyone. So like Dean Mooney said, my name is Gavin Gonzalez. I'm a second-year PhD student in biomedical engineering, and I'm co-advised by Drs. Shiny Varghese and Stefan Zoucher. Today, I'm going to talk to you about two of the research projects I'm working on. The first being a cartilage binding boundary lubricant, which allows lubricants in the knee to bind the cartilage. And the second project is a join on a chip ex vivo platform, which sort of allows us to understand how the lubricants interact with the cartilage in the knee during motions similar to walking around day to day. So it's important to understand first that the cartilage in the knee is constantly moving. As we walk about every day, the joint is very mechanically active. And the synovial fluid, or this yellow part of the image, helps promote the movement of the cartilage smoothly. Major issues with this are that injuries to the joint can cause cartilage degradation and altered lubrication, which can lead to the cartilage to wear down more and possibly even pain. And so treatment for this is exogenous lubricants can be injected to augment the lost synovial fluid to help promote better lubrication, allow cartilage to degrade less, and have less friction. So like I had mentioned, the lubricants control seamless joint movement, and hyaluronic acid is key to biolubrication throughout the body. Hyaluronic acid is a polymer that I showed on the bottom part of the screen here. Schematically, it's this squiggly line, and chemically, it's just a lot of these saccharide units. But the main quality of them is that they have an oxygen that's negatively charged, which allows them to be dissolved into a fluid and function as a good lubricant. So hyaluronic acid interacts with a lot of the other molecules inside the knee joint. So as you see on the bottom right-hand side of the screen, there are these green molecules called lubricin. And lubricin binds to the cartilage, and it also binds to hyaluronic acid. And this interaction helps to anchor hyaluronic acid to the cartilage surface. And when hyaluronic acid is close to the cartilage surface, as during mechanical motions, it makes it a much more effective lubricant to protect the cartilage. So what I want to do for my research project is to design hyaluronic acid so that it can bind to the cartilage on its own. And I want to do this by using electrostatic interactions. So in order to do this, I'm leveraging the fact that the cartilage extracellular matrix is negatively charged. And so I'm going to design positively charged molecules that can penetrate the cartilage. So for this, I'm using highly branched polylysine polymers, or HBPLs. Schematically, I showed them as these green squigglies. I believe that they will go into the cartilage on their own. So what I'm going to do is I'm going to conjugate them to hyaluronic acid. And what this should do is it should anchor the polymer to the cartilage surface, almost acting like putting little hooks on the polymer. So this is very similar to lubricin in the cartilage. And again, the reason why I believe this will work is because the HBPLs are highly positively charged. And so designing a lubricant requires us to understand how the lubricant interacts with the cartilage. And in order to do this, I want to devise a device so that I can understand how the lubricant behaves during biomechanical interactions as we walk around every day. So I'm trying to model joint movements like jumping, walking, or running in a joint on a chip device, so that in these motions, I can understand how the lubricant interacts with the cartilage. Readouts I'll use for this is to understand how much the cartilage is degrading and understand the coefficients of friction that the cartilage experiences during all of these motions. So thank you, that's my presentation, and I would really like to thank both my labs for helping me with this research.
Gavin, thank you. This is very important research for those of us who are in a different season in our lives. I'm hoping you find some really great answers for us. Thank you so much. Christina Hansen, chemistry.
Okay, so hi everybody, my name is Christina Hansen. I'm in the chemistry department in the Wong lab, and my research project is titled Apomorphine Derivatization for Dopamine 1 and 2 beta-arrestin bias ligands. So dopamine is a neurotransmitter that's present in our brain that's responsible for a very wide variety of functions, such as motion, the reward system, and cognition. And it acts on the dopamine receptors 1 through 5. And more specifically, dopamine 1 and 2 are involved in locomotion, which will be more important later in this talk. If we look in the figure on the right, this is a GPCR, which is a type of receptor that dopamine receptors are. And they signal through a couple different pathways, such as the G protein and the beta-arrestin pathway, which I will touch on again later. And you could go ahead and advance. And dopamine receptors have implications in several different neurological diseases, such as Alzheimer's, schizophrenia, and Parkinson's, where Parkinson's is my subject. So this disease is characterized by the loss of dopaminergic neurons, or neurons that produce dopamine. And the loss of dopamine causes bradykinesia, which is the tremors that you often see Parkinson's patients with. It also causes involuntary movement and rigidity. And L-Dopa is currently the gold standard drug that's been around for several decades now, and people often ask why you can't just inject Parkinson's patients with dopamine. But the reason for that is because dopamine cannot cross through the blood-brain barrier, and which is essentially a film that's on our brain that prevents drugs from entering. And if we look at this scheme, we see that L-Dopa is on the left, and once it becomes decarboxylated, which means the release of carbon dioxide gas, it'll release dopamine into the brain. And 90% of L-Dopa gets decarboxylated outside of the brain, which is a very huge issue if you advance. And there are several different complications with L-Dopa, such as poor bioavailability. It's also very susceptible to the metabolism, and because of these 2 points, requires a high dose for therapeutic effect. And these 3 points result in L-Dopa-induced dyskinesia, where dyskinesia is another form of tremors. And it's been found that chronic use of L-Dopa causes LID in 100% of Parkinson's patients after a decade. There's a very clear need for further development in this field. And if you advance, apomorphine is another FDA-approved drug for Parkinson's. And if we look at the 3 structures on the top, we see that they have some commonalities. So they have a catechol, which means the ring with the 2 alcohol groups, and a 2-carbon linker with the nitrogen functional group called an amine. And we see that in L-Dopa and in apomorphine. If you advance. Apomorphine also suffers from poor bioavailability, and it activates all 5 of the dopamine receptors non-selectively, and this results in a very high dose being required for therapeutic effect. If you advance. And as I mentioned earlier, the dopamine receptors can signal through a couple different pathways, such as the beta-arrestin and the G protein pathway. And it's been found that the beta-arrestin pathway is tied to the therapeutic effect, while the G protein pathway is tied with the side effects. If you advance. And so we found that, or we think that it's a very strategic way to treat Parkinson's through selectively activating the beta-arrestin pathway. So my plan is to activate or to create a ligand that would be biased for the beta-arrestin pathway at the dopamine 1 and 2 receptors. If you advance. And the way I'm doing this is through derivatizing apomorphine at these 3 areas that are shown by these circles on this structure. And so by changing one piece of the molecule at a time, this is called structural activity relationship, you can learn a lot about what portion of the molecule is important for the biological activity. And I'm doing the synthesis here at Duke, and we're sending these derivatives off to our collaborators at the University of Florida, where they'll be evaluated for their bias of the beta-arrestin over the G protein mediated pathway. And that is it.
Thank you so much. Genesis Higueros, mechanical engineering.
Hello, everyone. Thank you for joining us. Shout out to my family for being here. Today, I will be discussing bio-inspired vascular pore structures for fast charging batteries. My research attempts to enhance the performance of batteries at fast charging rates. Why do we really care about charging rates when we can already charge our phones within an hour or two, right? Well, there are still some challenges when it comes to green technology. For example, the charging rates of electric vehicles do not compare with those of internal combustion engines. So we can gas our cars within 3 to 5 minutes, but if we want to charge an electric vehicle from 0 to 80%, that will take us at least 40 minutes. And so a 4-hour drive from Durham to Washington, DC may increase anywhere from 5 to 7 hours when using an electric vehicle. Okay, so fast charging and more energy-packed electrodes are necessary for batteries at fast charging conditions. But what is limiting our ability to fabricate such devices? Well, to begin, batteries degrade at fast charging rates. We can control the current passing through a battery, increase the electrochemistry within, but this will not do us any good if the lithium ions are unable to intercalate into the electrode. Now, to provide some clarity, in the middle, we have a schematic of a 2D electrode in which, right here, in which the lithium ions are attempting to penetrate the electrode during charging. However, as one can observe, the lithium ions are limited to the surface of the electrode and don't utilize the material to its maximum. And so 3D electrodes, the schematic on the right, create paths for lithium ions, and in theory increase the penetration rate as a result of the increased surface area. Well, 3D electrodes have been made in the past and have proven to enhance the performance of batteries at fast charging rates. But their structures tend to be periodic or completely random. And so we have to ask ourselves, is this the best structure for the maximum performance we can achieve? What if we attempt to look for the best channel configuration that will give us the most efficient results? Well, we look to nature for inspiration and find vasculature in almost all forms of transportation in nature, right? We could see this in plants, in trees, corals, and even within ourselves, in our nervous system and our lungs. And so we hypothesized that vascularized channels will give us the best performances for batteries at fast charging rates. However, we don't know how many branches we need, how thick the branches should be, how far apart they should be, etc. We can try to fabricate multiple configurations and test each one out, but this will take us years to accomplish. And so if we can use neural networks to train a computer and that will predict the results of all of these millions of possible configurations. And by inverse design, choose the best porous structure. And to prove this, I will, once we have found that design, I will integrate that into our electrodes, test our battery cells at fast charging rates, and then compare with other 3D structures. And although I will be applying vasculature to batteries, this method can be integrated in a variety of applications. Here are just a few examples of how nature's vasculature has inspired innovative energy technology. Thank you so much.
Thank you so much, Genesis. Brian Kundinger, Statistical Science.
My name is Brian Kundinger. I am a second-year PhD student in statistical science, and I'm going to talk to you guys today about entity resolution. So entity resolution is a process of identifying common entities across separate files of records. So this is really easy when we have social security numbers or other highly reliable information, but it's really difficult when the data has a lot of errors in it. And this kind of mundane procedure has surprisingly important applications in areas like business, public health, human rights, and all sorts of things. And in one sense, I could talk about this as an unsupervised machine learning classification algorithm, but I'm gonna avoid some of the math to kind of talk to you about why I think this question matters a lot. So what does it look like? I have a bunch of data sets and I have a bunch of names in it. And I'm wondering who is the same person? Is Steve Feinberg in purple really the same person as Steven Feinberg over in red? And basically what I do in my model is I convert all the pairs of records into a comparison vector of 0 and 1s or 0, 1s, and 2s, indicating level of agreement in certain fields. So for example, Larry Wasserman to Steven Feinberg might be a 0, 0, 0. but a Larry Wasserman to Stanley Wasserman might be a 0-1 to indicate that I do have an agreement on this last name field. And then I put all those things into my model, and it helps me predict which pairs might indeed be a match. And again, this mundane procedure, it sounds really boring, but it ends up being important in a lot of applications. For example, sometimes people do it very, very badly. So in 2008, the Georgia legislature decided to make this exact match law, where if somebody came to vote and they didn't have the proper identification, they would fill out a provisional ballot, and every piece of information from that provisional ballot had to match the voter records. And it eventually was discarded because it was discriminatory against Black and Latino voters in particular. And mathematically, my critique of this system is that record linkage was deterministic. It had a very specific set of rules you had to match in order to meet the threshold, whereas the models that we're making are probabilistic, which are more robust to errors. So this is when it's done really badly, but also it can be done really great. So the DNC recently announced that they created a new national record linkage system, which has helped them increase their efficiency by 9%. Businesses and hospitals do stuff like this all the time as well. And another surprising application, my advisor, Rebecca Stortz, has used this methods to more accurately estimate the casualties in the Syrian conflict, for example. So my particular interest in this field comes in settings where the reliability of the fields and the rates of matching might vary by subgroup. So this occurs, for example, when you're matching records for women who change their last names more often than men. The last name is less reliable for women than for men. Addresses are less reliable for young people than they are for seniors. And all this stuff is really important, not only for matching the records themselves, but also for conducting downstream analyses. If I'm going to collaborate with a social scientist and they need to merge records, if they haven't properly accounted for these discrepancies in the record linkage process, then their downstream analysis can be biased. So this is what I'm working on, and thank you for your time.
Thank you so much, Brian. Now I want to ask everyone to briefly unmute, and let's give this first round of presenters a big hand. Can you unmute? Let them hear you clap. Thank you so much. And I also want to acknowledge the family members who have joined us. We are so proud of your children, and we are so honored to be able to join them on this journey as they pursue the PhD. So thank you so much. We're going to take a 3-minute stretch break. It's 3:38. You do the math. So everybody mute, and everybody turn off your video for 3 minutes. Take a break. Thank you, guys. Okay. Next up, it's Ethan Los Cesaro. Did I get that right, Ethan? Cesaro?
Yes, you did.
All right, your turn.
All right. Hi, y'all, I'm Ethan, and I'm going to tell you about my research, sparse conic H2 control. Now, this is going to get pretty abstract, so let's start off with a fun example. Say you want to shoot a rocket to the moon. You know what trajectory you want to be on. That's this set of positions and velocities through time on the green line. And you have a model of the system that says how your engine thrust maps to position and velocity. Your first thought might be to solve these dynamics backwards and figure out a priori what thrust you need at each point in time to move on this trajectory. So you could do this, but what happens when the wind picks up? It's not in your model. It's going to start to blow you off course. Now your engine thrusts are all wrong, and well, yank into the moon. So in practice, you hook up the rocket with sensors that tell you where you are relative to your trajectory, and you feed those measurements through a set of equations called a controller that adjust your inputs accordingly to get back on track. Control theory is the math behind designing this controller. Now, I don't work with rockets or really any other specific system for that matter. In control theory, we deal with a very general mathematical concept of a system. This way, the results can be generalized to any number of systems, from your espresso machine to robotic systems, but also non-mechanical systems like chemical plants, electrical systems, biological and anatomical, even social, environmental, and economic. However, one system in particular that motivates my research is a power grid with distributed renewable energy generation. In particular, I'm interested in a few key challenges that this type of system presents. It's subject to random disturbances, it's difficult to accurately model, and it has subsystems that need to communicate across huge distances. I'm going to take you through how my research tackles each of these challenges. First, let's look at disturbance rejection. We assume the disturbance is an unknown random noise process, and we think of the output of our system as the error relative to where we want to be and how much control effort our controller put in to get us there. We want to be able to handle a lot of noise with as little effort and error as possible. The type of controller that does this is called an H2 optimal controller, and for a simple enough system model, this is actually a solved problem. However, models lie. In particular, mathematical assumptions we have to make for our H2 optimal controller are often too restrictive. So even though the H2 optimal controller performs well on our model, it might not actually guarantee the stability of the true system. So we need some way to ensure that our controller is robust to modeling errors. My research approaches this with conic sectors. A conic sector, visualized in these plots, actually describes a set of systems based on how their inputs map to their outputs. So we can plot a system's input-output pairs on this top plot. Any system that lies entirely in the shaded region is called in the cone from A to B. This is a really flexible plant description. It can describe nonlinearities, time variance, parametric uncertainty, more or less all the things that our H2 model couldn't. And so it's a lot closer to reality. We also have a theorem that says if the system is in this cone, any controller in this other cone will stabilize the closed loop. So for robustness, we force our controller to be in the appropriate cone. The last design challenge is communication costs. Suppose we're looking at the electrical grid just over North Carolina. All of these cities are physically connected to one another, so traditional controllers require every measurement to be communicated between every subsystem. This is really expensive to implement, and it can also erode performance and stability because long distance communications can introduce unwanted dynamics and severe delays. So we want to design a sparse communication structure like on the right, while still guaranteeing good performance and stability. Each component of this problem has been solved independently, but all three have never been solved together because of their overlapping mathematical challenges. My approach is to reformulate this problem as a series of easier problems that converge to an approximate solution. In fact, just a few months ago, I published my first algorithm for doing this, and I'm currently working on improving these results in a number of ways. It's my time. Thanks for listening.
Thank you so much, Ethan. And I apologize, I forgot to say that you were in mechanical engineering. Next up, Ezine Nwanko, statistical science.
Hi, everyone. Thanks for being here today. My name is Ezine, and I'm going to be talking about methods for mitigating unfairness in machine learning. So for a long time, we believed that data was unbiased and objective, and so were the models and algorithms built on them. But as it turns out, that's not always the case. So in a typical data science pipeline, we start with a bunch of data about individuals, their behaviors, and their personal history. At least in the context that I look at social data. And we feed that into a machine learning or statistical model, which can look really simple or sometimes extremely complex and uninterpretable. And as a result, we get predictions that are then used as decisions or used to inform decisions about people and increasingly consequential decisions as it relates to criminal justice and loan or financial systems, as well as hospital situations. And so, for example, in 2017, journalists at ProPublica did an analysis on a proprietary algorithm that saw that Black men were twice as likely of being miscategorized by the algorithm as high risk for committing another crime compared to their white counterparts. And so what's being done to address these issues that we've termed algorithmic bias? So we can categorize the current literature into three main mitigation strategies where they kind of match where potential biases tend to creep in. So there's pre-processing, which says, let's do some sort of transformation to the data before we even begin modeling. The in-processing says, let's change how we estimate this model. And post-processing says, let's transform some of the results after the modeling. And all of this is being done in accordance to some predefined measure of fairness. There are many conflicting definitions of fairness that we won't get into now, but it is a really hot topic to figure out which fairness definition to use. And so my current research tries to take methods from causal inference to address these issues. We try adding a constraint to a model that basically says, let's make sure that the expected difference in outcomes between individuals, say Black and white individuals, is zero or as close to zero as possible. And this constraint uses a method called overlap weights that basically we like them because they're robust to any unmeasured dependencies in the data, which is nice because social phenomenon are not always measured and they're also not always available. And so you want to, as a modeler, you want to make sure that you don't just ignore their existence just because you don't see them. And then for post-processing, we try to propose estimating what we call a fairness adjustment that we essentially add to our distribution of predicted outcomes for the underprivileged group to make sure that their risk distributions are independent of a predictive variable. Again, think race or gender here. We want to kind of make sure that they're similar. And lastly, I would like to close with what my research goals are. So often people ask if I believe that fixing these models and data will solve some of these tough societal challenges. And while I don't believe that these models alone will magically fix everything, I do believe that computing has a valuable and supportive role to play in pushing for social change. Specifically, there's a really nice paper that kind of outlines these 4 roles as a diagnostic to diagnose when these issues arise, to formalize what exactly we mean by certain policies and issues, as a rebuttal against certain policy efforts that aim to use computing towards social change, and also as a synecdoche, which is a new word that I learned, but it basically is to shed light on new light on an old social issue. And so I aim to have my work play one or more of these roles going forward. So yeah, thank you to my advisors and to all of you for listening. And I'd love to chat more in the breakout room if you're interested.
Thank you so much, Ezine. Alexander Mangus, Electrical and Computer Engineering.
Thank you everyone for coming today and listening to my talk. I'm Alexander Mangus, a second-year PhD in the Electrical Engineering Department under Dr. Aaron Franklin. And my research is in high-performance nanoelectronics, namely 2D transition metal dicalcogenides to go beyond Moore's law. So for some background, Gordon Moore was a pioneer in the transistor industry. In 1965, he published a paper where he introduced Moore's law, which said that as long as it was cost efficient, the number of components on a chip will double every 2 years. Essentially, he was predicting the advancement of digital electronics and computing power. And this trend held true for 60 years based off of 2 things. One of them was the dominance of silicon in the transistor industry and the continuous scaling down of transistor size. However, the dominance of silicon is close to coming to an end due to physical limitations of silicon in transistors at current sizes. So this graph shows several issues facing modern day transistors. The first of which can be seen in the inlay in the top left, outlined with the black border. And this shows that it's actually no longer cost efficient to continue to add transistors to chips due to the increased device complexity at these small sizes. The second issue is seen in the blue oval highlighting the teal line. And that shows that maximum clock speed has essentially peaked. So even though we're adding more transistors, due to the physical limitations of these transistors, their device performance has stopped. So adding more does not increase the maximum clock speed. And the last one can be seen in the bottom oval, the gray line, and that shows the thermal design power, essentially the energy and heat that is being generated by these chips. So we would like the energy to scale down along with the sizes of the transistors. However, that is not what we have seen. So by adding more and more transistors, we're exponentially increasing the energy drawn from these chips. So with the end of silicon in sight, scientists have turned to 2-dimensional materials. And one such subset of these 2-dimensional materials is 2D transition metal dicalcogenides, which is what I work on. And these TMDs can go down to less than a nanometer thick, which allows them for smaller devices. And they also have reasonable band gaps and comparable electrical characteristics to larger silicon devices. And that gives them applications in not just digital electronics, but also biosensors, energy storage, photonic devices, etc. And some common TMDs are molybdenum disulfide, tungsten disulfide, and tungsten dicellulite. So one of the main challenges facing these materials is a high contact resistance, essentially a barrier to current injection that stems from their pristine surface. So I work on modifying these contacts to allow for a higher on current in these devices. So my research specifically is an ion beam modification in an ultra high vacuum chamber, which can be seen pictured here. So we bombard the flake with ions, which creates vacancies on its surface to allow for better contacts. So in conclusion, while Moore's law is ending for silicon, these two-dimensional materials could push us to continue Moore's law or even go beyond.
Thank you, Alexander. Grayson Rice, Biomedical Engineering.
All right, thank you very much. Hello, everyone. My name is Grayson. I'm a second year in biomedical engineering in the Gersbeck lab, and today I'll be talking to you about regulators and cofactors of neuronal maturation. So as a brief introduction, there are over 6 million people in the United States alone suffering from neurodegenerative diseases such as ALS, Alzheimer's, or Parkinson's disease. This number only continues to increase. And as a result, there's a need to develop in vitro models to study these diseases, which means looking for ways to study them in the laboratory setting on a plate as opposed to in a live person. But the problem is that primary neurons are very difficult to obtain and study because you can't take brain tissue from a living person, and getting neurons from post-mortem brain tissue is also very difficult. And so the solution to this problem is to generate neurons from stem cells using a method called transcription factor reprogramming. And so taking a step back to the basic biology, transcription factors are proteins responsible for gene expression. And so in this figure on the right, you can see you have gene X that makes transcription factor X that goes back into your DNA and binds to other genes, either turning them on like A through C and E, or turning them off like gene D. And which genes are turned off and off is responsible for your cell identity. And this is why your skin cells are different from your muscle cells, even though all of your cells in your entire body have the same DNA. And so what people have found is that by overexpressing certain transcription factors, you're able to promote neuronal differentiation. And so in this figure, you can see how by adding a variety of different transcription factors, you can go from pluripotent stem cell to a different brain cell type, like a neuron, oligodendrocyte, or astrocyte. And then here's what differentiation actually looks like. So compared to the top row, which is just stem cells, adding a transcription factor to stem cells will get you neuronal looking cells within 4 to 7 days, which is a really powerful technology because stem cells are self-renewing, so you can have an infinite supply of them. And in effect, it means you can have a potentially infinite supply of neurons that you can generate in a week's time. But this method does have its drawbacks. One of which being that these transcription factor-induced neurons lack functional maturity. And one way that you can measure that is with the action potentials of these neurons. So if you just induce neurons with one transcription factor, you get pretty weak action potentials, which is similar to fetal or baby neurons. And so when you're studying late onset diseases like Alzheimer's, having neurons that resemble those of children or babies isn't really a reliable model. But what people have found is that when you express two different transcription factors in combination, you can get more robust action potentials, which is more akin to adult neurons. And so what I'm looking to do is to identify the pairs of transcription factors that promote neuronal maturation by co-activating NeuroG3, which is a transcription factor known to cause neuronal differentiation, along with all other 1,500 transcription factors in the human body. And here's what that approach looks like. And here are all the technical details, but what this diagram really boils down to is to overexpress the transcription factor pairs in stem cells such that each cell only gets one combination. You let them differentiate, and then you identify and sort the mature neurons from the immature neurons, or the cells that don't differentiate at all. And then you recover their DNA to find successful combinations of transcription factors. And with this knowledge, we hope to develop more reliable neurodegenerative disease models to potentially cure Alzheimer's, ALS, or Parkinson's using more mature neurons. Thank you.
Thank you, Grayson. Gianna Cetone, Chemistry.
Hi, my name is Gianna Cetone, and I'm a second-year grad student in the Becker Lab here at Duke University. And today I'll be talking about my research on fully resorbable stereocomplex linked hydrogels with tunable mechanical properties. So before I really delve into my research, I would like to talk to you about human mesenchymal stem cells, which Grayson actually just touched upon in his presentation. So human mesenchymal cells have the special ability to become other types of cells, and we call this differentiation. Due to this ability, human mesenchymal cells show promising applications for tissue engineering, such as being used for bioprinting. But the real question is, how do we control this differentiation? Recently, it was discovered that differentiation can actually be controlled by altering the rigidity or modulus of the microenvironment that they're grown in. It is well known that different tissues have different moduli. For example, bones are much more rigid than the brain. So scientists discovered that by growing human mesenchymal cells in synthetic hydrogels with the same modulus as native tissue, that they can actually direct differentiation towards that tissue cell line. So scientists have been working on creating a system in which you can change the modulus by using small changes to the gel chemistry or solution and create a range of moduli that will grant them access to creating or generating most of the cells found in the human body. So while some scientists have achieved this goal, the networks that were formed using their techniques created chemical bonds that are extremely hard for the body to break down if these gels were to be implanted. This ultimately prevents the translation of their gels to research. So with that being said, my goal of my research here at Duke is to create a gel system with not only a tunable modulus, but also a completely resorbable hydrogel network. Thus, this material can be implanted in the body, do its job, degrade, and then the body can get rid of it. But to do this, I will form a network comprised of a hydrophilic degradable polymer known as polyethylene glycol, or PEG. This is seen in black here on the slide. And then these four-armed PEG stars will be N-functionalized with a polymer called polypropylene succinate, seen in red and blue here on the slide. And this is where the cross-linking will actually come together to form the gel network. And this cross-linking will occur using an interaction known as stereocomplexation. So stereocomplexation is a physical interaction between two polymers of opposite orientation, and it practically binds them together really tightly. Polypropylene succinate can exist in two orientations, R or S, and this means that they have the same chemical configuration, but they're just mirror images of each other, kind of like your left and right hand. And by functionalizing the PEG stars with these S and R polypropylene succinate strands in solution, the network can be assembled through a formation of a stereocomplex without forming any actual chemical bonds. And then by altering the lengths of PEG and polypropylene succinate, we should be able to alter the network density and modulus of the gels without forming any chemical bonds that could inhibit degradation in the body. In conclusion, with the completion of this project, this material can then be implanted for the regeneration of human tissue with no need for surgical removal like previous gels. Furthermore, this material can even be seen in applications such as bioprinting or manufacturing of artificial organoids. Thank you so much, and now I'll let Eric close us out.
And Eric's in electrical and computer engineering.
So I'm Eric Yates. I'm advised by Dr. Hiley in the ECE department. And today I'm going to be talking about my recent work on improving machine learning security with complex valued neural networks. So this builds on deep learning, which is the process of learning a hierarchy of abstract concepts towards achieving a high level goal. And nowadays that's achieved with very deep neural networks from tens to hundreds of layers deep. And we really like these because they're high performance. They're used as a building block for intelligent systems such as autonomous vehicles, drones, medical and imaging and recommendation systems. But there's a problem with this in that it's really easy to create these things called adversarial examples if you know a little bit about the training procedure of deep neural networks. So you can craft adversarial examples by taking a normal input, X, which is classified as a panda. Then you add a small amount of carefully crafted noise to this image, which still makes it look normal to us, but it looks very different to a machine learning classifier. So now it thinks it's a gibbon with 99.3% confidence. And so adversarial examples are typically generated using the gradient of a network's output with respect to its input. And this can be a really big problem because you can actually create adversarial examples in the real world. So imagine that you have a Tesla or an autonomous vehicle that's supposed to be able to detect stop signs. Well, If you know how to create adversarial examples, you can create an adversarial sticker that causes that machine learning model to not recognize the stop sign. And another big problem with these is that adversarial examples are transferable. So if you craft an example that works for one network, it will often work for other networks in the same way. So there's some common security techniques. The one that's most well known is adversarial training, which is basically to incorporate adversarial examples into this training procedure. And it's the most effective in practice, but there are some issues with it in that it requires prior knowledge of an attack, and training is very expensive. So I've been working on improving this method called gradient regularization, which relies on the idea that making loss gradients small should make gradient-based attacks more challenging. So this is a good method because it's scalable to very large networks, and it's also independent of a specific attack model. But there's a problem with it in that if you want more security, you need more gradient regularization, which decreases performance on standard objectives such as stop sign detection or image classification. So in order to understand this issue a little bit better, I'm going to talk about training deep neural networks. So in general, you'll define a loss function in which there's a loss surface and minimal points of this loss surface coincide with good performance on the desired objectives, such as image classification. And so you use gradient descent to guide your machine learning model down into this local minimum. Now, when you add gradient regularization, this adds in extra objectives for the machine learning model to complete. And so while this machine learning model achieves these other objectives, it might not achieve as optimal a solution on the desired classification objective. So what I do is I design a complex value neural network, which leverages complex value computation to help with this gradient descent process. And it's a lot like standard neural networks, but it includes this constraint during the gradient descent process, such that if changing a neural network parameter is important for one objective, say image classification, then that same change must necessarily be unimportant for the other objective. And so when we train these complex value neural networks with gradient regularization, they're able to achieve more secure outcomes and also perform better on the standard objectives, such as image classification. And so we can compare the accuracy of these different classifiers as we tune up the strength of an attack. And so the black line is the adversarially trained network, which is trained with prior knowledge of the attack. And then the blue lines are the standard neural networks, which are trained with gradient regularization. The red lines are the complex value neural networks, which are trained with gradient regularization. And we find that the complex value networks are able to achieve a security level comparable to that of adversarial training, which has knowledge of the attack, and are able to achieve higher accuracy than the standard neural networks. And so these results are reproducible on multiple data sets in different settings. And by doing this research, we hope to improve neural network security methods, which are both scalable to very large networks, and which are independent of a specific attack model. So thank you for listening to my talk.
Thank you so much, Eric. Thank you. So I need everyone to unmute and give these incredible scholars a big round. Thank you so much. The quality of your thinking and the things that you're about to do is just so amazing. And guess what? That was 13 presentations in 52 minutes. Maybe we're on to something here. Maybe we're on to something. Thank you. And now I'd like to introduce Dr. Jennifer West, who will introduce our keynote speaker, Erika Moore. Jennifer West is the Fitzpatrick Family University Professor of Engineering and Associate Dean of PhD Education at the Pratt School of Engineering. She has been elected to the National Academy of Engineering and the National Academy of Inventors. She is a co-founder of Nano Biosciences. Dr. West's lab alumni are leaders in both academia and industry. During her career, Dr. West has trained over 45 graduate students and over 23 postdoctoral fellows. Her mentorship emphasizes career development and the importance of fostering diversity and inclusion in the science community. Dr. West has received numerous accolades throughout her career, including the 2016 Capers and Marion McDonald Excellence in Research and Teaching Award in the Pratt School of Engineering. She has also received the Graduate School's 2019 Dean's Award for Excellence in Mentoring. Dr. West.
Thank you so much, Dean Mooney. So I must say that probably one of my proudest accomplishments is serving as a mentor to our next speaker, Erika Moore. So Erika received her PhD in biomedical engineering from Duke, and she worked in my lab. She did really groundbreaking work to understand how a set of cells called macrophages influence the development of new blood vessels. And to do this, she built an engineered system that gave her exquisite control over the types of cell matrix and cell-cell interactions that were occurring, and allowed her to visualize in real time how these interactions took place. So she had wonderful publications, really brilliant science done here at Duke. She then went on to postdoctoral training at Johns Hopkins to further her knowledge of both engineering and immunology. And then this year she started a faculty position at University of Florida. And I certainly don't envy her having to start in pandemic conditions, but she's done a remarkable job already teaching courses in biomaterials, getting her research lab going, already having success in grant funding, and really doing remarkable things. Despite all of her scientific brilliance and her wonderful contributions, I think one of the things that really makes Erika special is that she truly cares about the development of people around her and will do anything to help out students, colleagues. And one of her contributions, as an example of this at Duke, was to create a mentoring program to help sophomores in engineering make the transition into engineering, be successful in their majors, and thrive as future engineers. This was a point in the curriculum where we would see typically a lot of attrition of students, and especially women and underrepresented students. And her program has had a great impact there. And that's just one example of the way that she goes about making the world a better place with all of her interactions. So with that, I introduce Erika Moore Taylor, and I'm looking forward to her talk. So thank you.
Thank you so much, Dr. West, and hello, everyone. It's really nice to be back at Duke. I was asked to kind of share my journey and perspective through the PhD and to make it applicable. So being the person that I am, I came up with a list of action items that I thought would be helpful to share with you, and I think they can be applied at any stage of your training. So this talk is titled Journey Through Your PhD. And I want to start by humanizing myself. So my name is Erika Moore Taylor, and as Dr. West said, I graduated with my PhD in 2018, which feels like a world ago. I had a lot of friends, community, family that I relied on throughout my PhD. And so I think, you know, even by looking at these photos, you could tell this was all of the images were taken pre-COVID-19 pandemic. Also, that I had a lot of fun and I had a lot of support throughout this journey. But something that's not as readily mentioned or appreciated is that I struggled with my mental health and feelings of burnout. Sometimes I felt isolated. I questioned the role or purpose of the PhD. And so what I hope to share today is a message of resilience paired with self-care. And that is kind of what allowed me to finish my PhD and to go on and be quote unquote successful. So I want to start also by saying that you've got this. If there's any hesitation, doubt, concern, I'm here to say you have the ability within you right now to complete your PhD or to complete whatever training program you're in. It's really just a matter of how and when, not if. So take that message of encouragement with you. Understand that you have the innate ability to finish your PhD. One tip and question that I often ask myself is, how would your behavior change if you knew that you have the ability to get a career you enjoy and graduate with your PhD? So in the future, if you knew everything afterwards was going to be a-okay, how would your behavior change your current, your present? Oftentimes answering this question can be challenging because you're like so in the weeds, you're so worried about all of the things that you have to do and how that will play out in your future. But instead of kind of foreshadowing failure onto your future or foreshadowing, difficulties onto your future, know that your future is kind of secured and focus on making the best out of your present. And so I'm going to share with you how I did that and how I embraced this message. So this presentation is going to be filled with tips and advice, walking you through a PhD. I want to throw out the caveat that this is a sampling size limit to an N of 1. So I'm just sharing my perspective, but I hope these are tips and tricks, maybe advice that you can apply. And like I said, there are skills that I apply still to this day. One thing that I also want to ask as we get started is to think about what areas do you feel in need of help? What areas do you feel like, yeah, it's not quite going as smooth, especially with the pandemic and being mostly virtual? Are there specific areas where you need additional resources or support? And hopefully by identifying them, we can tailor your comments or even in the breakout rooms to address some of these concerns. Okay, so when I first was invited to speak, I went back through and looked at all of my images and photos year by year throughout my PhD. And what I've noticed is that this is really a journey of growing in confidence, becoming more accustomed to setbacks, and staying focused. So I made these 5 categories, these 5 action items that I think really significantly helped me throughout my PhD. We're going to go through each of them, but to orient you to the entire list, we start by talking about what's most important, creating a community, getting support, knowing your why, and being fearless. All of these techniques, I think, will help you become a better PhD student, a better PhD candidate, researcher, scientist, engineer, and can be applied to your projects, which were really cool. Okay, so I'm walking you through as myself. This was me when I started my PhD, my first year at Duke, and I had no idea what was most important for me to focus on or how to get traction in my PhD. So I often found myself distracted. I was working really hard, reading a lot of papers, trying to formulate my thesis project and address the questions that I thought I needed to address. But I found that I was pulled in lots of different directions. So I wasn't making a lot of progress, I felt, during the first semester of my PhD in particular. So this is where the first tenet came into play for me. And this tenet is called what is most important or most important. So I actually went to Dr. West and I said, Dr. West, this is going to be my goal this semester. Like, can I just make our hydrogel system with our blood vessels in it? And that gave me laser focus, essentially, to say, what is most important in this semester or this year? Each semester, year, you're going to have ups and downs in your PhD. That's a normal part of the process. But by setting a goal and framing it within the context of a semester or within the context of a summer or even a year, it allows you to stay focused on what's most important at this stage of your PhD. It's also important to know that progress is not linear. So one semester you can be like, gather all of this data, and then the next semester it could be like, figure out how to analyze all of the data that I've gathered. And I think it's okay to have the progress not be linear, but it's very important to clarify what is the most important thing for me to do this semester. So how I did this was by setting a priority. It's important to note that I said a priority is singular. So there is one important thing. If there is one thing that you can accomplish this semester towards your PhD, what is that? Write it down and try to work towards accomplishing that thing. Be aware of distractions. So there are a lot of times you think you can do multiple things really well, but when you're starting out in your PhD, it's really important to hone in and get focused on what's most important. Should I get this preliminary data that will prove, a hypothesis that we're trying to generate? Beware of distractions, because distractions also lead you to believe that you can multitask. And what I found in my experience is that it's hard for me to multitask well. if I don't know what my priority is. So by defining your priority, you know what's a distraction versus what's a part of accomplishing that first goal. Additionally, I really encourage you to ask your advisor. I met with Dr. West multiple times during that first semester because I had little to no idea what I was doing. And so you can seek their guiding oversight to basically say, okay, am I progressing through this goal? Do you think this goal is fitting of where I want my project to go? Seeking this feedback and getting all of this input will better help you be able to define what is most important. It's really critical to avoid distractions, and that's why I emphasize this one, because I think it is a foundational tenet of your PhD. These are pictures of me during my first year. Yeah, that's all I'm gonna say. The picture at the top was me when I first got to Duke, like literally the first day, and then going to some events around campus. So for me, Dr. West shared a little bit about my research, but what motivated me to even get my PhD was this fascinating field called tissue engineering or tissue regeneration. And this was the ability to kind of create a mimetic, a life mimetic, a biological replicate outside of the body of something that we wanted to maybe model or see if we could use to create new outside of the body. And so that's tissue engineering. It can be applied for model systems or to create entire new tissues that can be used in the clinic in the future. That was my why. But my why was also combined with immunology. So I became really fascinated with the immune system, and it's traditionally thought of as our defender. So if you get a cold, you want your immune system to help fight off that cold. But the immune system is quite plastic, and it can be used in a variety of different ways. So a burgeoning field within biomedical engineering is the application of the immune system with tissue engineering or tissue regeneration. And that's where my work came into play. Specifically, I wanted to focus on a cell type that was called macrophages. These are a part of the immune system, and they can be used to hopefully harness new tissue. So that became my thesis work. That became what I tried to prove and what I tried to demonstrate, and that became my focus throughout my PhD. We wanted to understand how we can utilize macrophages in tissue engineering. And so we tried to combine these cell types and investigate whether we could recreate our hypothesis, but within our controlled system, within our controlled polymer system. So that was awesome. I had focus, but after that, I hit another roadblock. So this brings the second tenet. So I had established my priority. I had established my first semester goal. In addition to classes, it was to create our blood vessel system. But I also discovered that I became lonely. So I started making progress, and this was intentional progress with the oversight of my advisor towards my PhD. But I found myself without close friends. And I actually think this is really important to emphasize, especially in the age of COVID-19 and things being virtual. And so there's the absence of close friendships. And so at least in-person, interpersonal friendships. And so the second tenet was to create community. And this is one of my favorite things because I think the onus is on me to create my own community, which means for you, the onus is on you to create your community. So yes, you create it. It might not exist in the shape or form that you want it to, but you have the ability to act and influence it. So I want to start by emphasizing that there is a new reality that we all exist in today. All the images here, which were super fun, taking during my PhD, that's not an option in this world that we exist in today. And so while virtual is not ideal, I encourage everyone to embrace the change. So what does this look like? To me, this looks like randomly asking people who I actually went to grad school with if they want to have a happy hour, or engaging with people who I've never met before to say if they want to play Among Us, for an hour or two on Saturday. There are lots of different ways to continue to engage community. Maybe it's starting a book club. Maybe it's listening to the same podcast and then commenting about it in a WhatsApp group. I think community is really important because it provides that infrastructure for you to have positive and hopefully positive interactions with others outside of your PhD. I encourage you to join student groups. So a lot of student groups at Duke are still meeting, albeit virtually, but I recommend try to find one group to join, one person to connect with, and then build your network from there. Additionally, the culture in the lab is yours to create. So I strongly encourage you, take the time to get to know your lab mates well. Ask, if they want to have a happy hour playing games after lab meeting. Be intentional about crafting community, because it's truly going to be the difference between a healthy, positive, resilient PhD or the absence of that. So with COVID, take it all in and still try to act and do something that will bring you joy. I try to do this at least once a week. Okay, so this is where we transition, because of course I have lots of friends, quote unquote, and I had started making intentional progress in my PhD. I felt like the work was going well, but then I encountered burnout. So this is something that I think a lot of people can relate to now, especially in the pandemic. And so I found myself with community, with a group of friends. I had direction within my project, but my motivation started to wane. So what happens when your mental health declines, when your experiments continue to fail over and over and over, and you don't know why, and you're at a loss for what to do next? I ask you to think about what are some healthy ways you can cope with exhaustion or with burnout, because these are going to become issues that you're going to encounter or have already encountered. So think about healthy ways to cope with them. This is where my third tenet comes in, and that's titled Get Supported. So there are a lot of resources at Duke, and here are a few of the ones that I took advantage of to address some feelings of burnout and mental exhaustion that I felt during my PhD. I visited the Counseling and Psychological Services, CAPS, and because I truly believe that my mental health, my emotional health, my physical health is tied to my wealth and my ability to successfully complete a PhD. So I recommend, if you feel issues with burnout, if you feel mental exhaustion, reach out to some of these resources. Additionally, I got heavily involved in the Graduate and Professional Student Council. So this was one of my prior student groups that I got and connected to create my community. But they also served as a resource to kind of talk about frustrations, talk about issues that I was encountering with my work, and get their advice for how to address them or how to create space so I could think and readdress or reapproach some of those issues. One of the other things that I truly love about Duke are all of the centers that they have. So I was in the Mary Lou almost every Friday. I know that that's not physically possible today, but they still host events. And I know, because I still get some of the student club invites. There's also the Women's Center and the student affairs. So there are a lot of additional resources, even if it's not counseling and psychological services, that will really help you get supported and will be the difference between you navigating successfully through some of the mental and emotional challenges of your PhD. Okay, so then, I was starting to get support, and this was around year 4 or so. And you start to have kind of an existential crisis. And so for me, this was, oh my goodness, why did I commit to my PhD? I used to call my father about this all the time. But a younger Erika would say, it seemed like a good idea at the time, I don't know. An idealistic Erika would say, oh, I really love macrophages and their roles in tissue formation. And I think they're such a cool cell to study. This is the coolest project in the world. A pessimistic Erika would say, this was the worst decision I ever made. Why did I make it? And so I think it's really important to ask yourself these questions, but to answer them. And so that brings me to my fourth tenet, which is knowing your why. This tenet is one of my favorites because it requires a lot of introspection and honesty. Why do you want this? Why do you want to study it? How does it impact you? One of the ways that I answered this question and answered my why was doing lots of personality assessments. So this is very fun to me, but basically trying to understand what in my personality contributed to me being here. Why did I commit to this? Additionally, you can ask family and friends who know you pretty well, what do I do well? Why did I think this was a good idea, you know? And they can kind of remind you, especially when you're going through some of the challenges that are inevitable in a PhD. Additionally, I really recommend you find positive reinforcements. Some of the people on this call, including, you know, my own advisor, but also other administrative staff, were these positive reinforcements for me. They would say, good job, Erika. Keep going, Erika. You're doing great, Erika, even when I personally did not feel like that was true. So find mentors, connect with them, trying to present in different shapes or forms. There are lots of virtual presentations happening today, and it's just nice to get out within the community and get some positive feedback on your work. Additionally, I had mentees, and so it was really fun to have undergraduates or medical students in the lab and have them kind of be training under me and emphasizing why this work mattered and why they were cool about it. The great thing about being excited about your work is, or is being around people who are excited about your work, is it kind of ignites that same excitement in you. So on those days when you're feeling a little bit less motivated, having an undergraduate come in and showing them how to do something, that can excite you and can really reinforce why you selected this path. Okay, so the outcome of my PhD was so fun, and using some of those 4 tenets that I talked about, I was able to build this kind of really robust community that propelled me through my PhD. It certainly was not me alone. And so at the end, we found that macrophages, they interact and they support blood vessel formation. And this is usually in a function dependent manner. So depending what function they're in, they can be more or less supportive of us forming new blood vessels in our tissue engineered scaffold. And that was really cool because that was what we set out to first prove or first demonstrate. But this brings me to my last and 5th tenet, which is to overcome the notion of normalcy. So I love this quote, and it says, overcome the notion that you must be regular because it robs you of the chance to be extraordinary. My last tenet is be fearless. And I'll talk about this internal locus of control, but one thing to consider as you navigate through your PhD is that if you master your mindset, you've mastered the world. This is something I still try to apply and enact on, but I'll tell you why I think this is so critical. So this tenet, as I said, is called Be Fearless. And one really awesome quote that I use often is, what would you do if you knew you could not fail? So I have this image of me as a little girl. I was really fascinated with biology, but it was never in my wildest dreams to come to Duke. This photo down here was actually taken during our recruitment visit when there was a snowstorm and we had to go to Whole Foods. And this is me, an undergraduate, right before I started grad school. And then the lower picture is me today. But what's not seen on this slide there was a lot of feelings of doubt and imposter syndrome, like, you know, will I be able to do this? Is this the right choice that I made? You know, how long is this going to take? Those kind of questions plagued me a lot. So that's why I started this presentation by saying, you've got this. If you trust in yourself, you've been able to navigate yourself to this place and to this time period. And so that's really critical because you can look back and say, wow, I've done so much. Can I trust in myself to do so much more? The answer is yes. I also want to encourage you to be courageous. Lean into some of your dreams. There might be problems that you think you want to solve that maybe you haven't begun to probe, or you haven't maybe done that one experiment because it's a little bit intimidating. I recommend that you lean in earlier rather than later. Additionally, I hope you can find your accelerators. So what makes your heart beat a little bit faster? Is it communicating your science? Is it getting data together for a publication? Is it, you know, trying to apply for different fellowships and supporting your advisor with grants? If there's anything that makes your heart beat a little bit faster, that's an accelerator for you. So usually it's a motivator. It's going to help you work a little bit faster and a little bit better. And I recommend you lean into that too. So that goes back to this internal locus of control. Be fearless is giving you the power to say, okay, how do I want to influence my life? How do I want to influence Duke University? When I first got to Duke, there was no way that I thought that I would be leaving as I did, being as involved as I did, you know, helping others as they were navigating their undergraduate, master's, or PhD. So by being fearless and applying the tenets that I've gone over, I was able to have that impact. But I wish I maybe would have been a little less, you know, hesitant or doubtful of myself. So that's going to conclude. I want to end by saying that challenge, triumph, repeat. So that's going to be what the PhD continues to be for you. You're going to you're going to be met with lots of challenges. You're going to triumph over them, and then you're going to be met with more challenges. So get used to that. By applying the tenets of identifying what's most important, creating community, getting supported, knowing your why, and being fearless, you will be well on your way to becoming an independent scientist and critical thinker, which is the goal of your training. I'll stop talking. Thank you.
Dr. Erika Moore, thank you so much. Thank you so much. So you all have to unmute and give her an incredible audible. Woo. Oh my God, Erika is so amazing. And I'm so thrilled that I was just a little part of that journey, watching you and watching Dr. West watch you. And I think that, Erika, you have just shown us how one can validate herself with purpose, passion, and power. Validate yourself, claim your greatness. And that's what she's telling us. Validate your greatness. Thank you so, so much. And now I'd like to turn things over to the person who thinks about, works on all things graduate education at Duke, Dr. Paula McLean. Dr. McLean is the co-PI on this Sloan grant. She's Dean of the Graduate School, Vice Provost for Graduate Education. She's the James B. Duke Distinguished Professor of Political Science and Professor of Public Policy. And she is our awesome leader in graduate education at Duke. Dean McLean.
Wow, what can I say, Erika? Welcome, welcome back, welcome home. You got to tell me about that Taylor bit, okay? I'm not sure exactly where that came from, but you can send me an email and kind of give me all the details. You know, it was such a pleasure today to hear from our Sloan Scholars, for them to share their research. As a researcher, it's critical to explain your work succinctly to people who are not experts in your field. And that is one of the reasons we developed the Duke UCEM Research Summit, so that our Sloan Scholars can have more opportunities to hone their skills in that area. I want to thank everyone in attendance today, and I want to especially thank our honored guest, Erika Moore Taylor, for coming back to tell us about the great things she's doing now. When Erika was, she kind of talked to you about her journey, but she left out that she was actually the graduate trustee on the board of trustees. So she was really actively involved. And even though when you look at her CV, you see Hopkins and Duke and now Florida, but if I remember correctly, you're actually from Colorado, right? Yeah.
Yes, ma'am. Yes, ma'am.
And as my fellow co-PI, Professor Howell mentioned in the opening remarks, a big part of Duke UCEM's work is about building a collaborative and a supportive community for our students. So I'm very heartened to see members of that community, faculty, staff, students, and alumni, but also I am just thrilled to see how many of your family have logged on to hear your research presentation. That is just fabulous that all of these people logged in today to support our Sloan Scholars. In just a minute, you will get a chance to do even more community building because we're going to have breakout rooms set up where you can chat with our Sloan Scholars and get to know them and their work even better. So please stick around and take part in those conversations. I also want to thank Professor Howell, Senior Associate Dean Jacqueline Looney, my colleagues in the graduate school, and all the faculty and staff who've contributed to the Duke UCEM. This initiative is a big undertaking, and without all of your tireless efforts, it would not be possible. So thanks to you, the Duke UCEM, administrators, faculty champions, everybody who works on this program. The Duke UCEM has had 3 very successful years and has more exciting plans in the future. And one of the things we've known is that we've identified through the UCEM process is that not only has the UCEM benefited the Sloan Scholars, but it's also helped us meaning the graduate school, enhance our support for other graduate students at Duke. And the Duke UCEM is serving as a model for other graduate student support efforts at Duke that the graduate school is undertaking. There is still much to do, but we also have much to be proud of as well. So I'll now hand it back over to Dean Looney, who will tell you about the breakout room sessions. And thank you again to everybody for joining us, and congratulations to our Sloan Scholars for your outstanding presentations.